sometimes we say things didn't scale well. We actually mean like it didn't make a good product.
Biotech doesn't fail because the science is wrong. It fails because the system around it breaks.
All right, welcome everyone back to another episode of Helical Brew, joined with my co-host Sura, Hadi. We were actually in the same zip code recently, weren't we, Sura? Right? I came down to San Diego for LRIG, and it was great to catch up. Had a good time there with a lot of our biotech colleagues and lab automation colleagues. But we are now back in our home spots for another episode of Helical Brew. And we're going to be joined with Rich Cohen, who's Senior Director of Datapoints at Ginkgo Bioworks. So welcome, Rich. And welcome to the show, everyone. It's great to have you with us. And welcome to Systems of Science, a special Helical Brew series exploring how biology becomes infrastructure and how real systems are built, scaled, and delivered. Today's guest is Rich Cohen, Senior Director of Ginkgo Datapoints, working at the intersection of antibody drug discovery, data, and product strategy. Rich Cohen has one of the rare vantage points in biotech, starting at the bench, moving through technical program leadership at Bolt Threads, and now helping shape product and platform strategy at Ginkgo Datapoints. He lived the transition from biology as an experiment to biology as a system, and now to biology as a product. Welcome, Rich.
Oh, great. Thank you. Great to be here. Thanks for the intro.
You bet. And we're glad to have you with us. And this episode is going to be titled, Biology Doesn't Scale, Systems Do. What it actually takes to build a biotech product. Always got to set the stage with that.
And as you can tell, we don't tell our guests what their episode's going to be titled.
Couldn't have done better myself.
All right, Rich, I think we talked about it. We love doing icebreakers at the end of the day. So here's our stab at an icebreaker. Rich, you've gone from running experiments to leading programs to shaping products. At what point did you realize biology doesn't scale on its own, that systems are what actually scale?
Whew, yeah, good question.
No pressure.
Deep tracks early in the show.
I think that's probably really at the transition when I was transitioning out of the lab into managing programs. And I was at Bolt Threads, and one of the founders there, Ethan Mirsky, was like, that thing you do where you pull people into a room and you get them talking about what they're doing, that's called project management, and we need to figure out how to do that. And you should bring that into have that process for that of how we do it, because we're now 30 people. I mean, just big enough where like not everybody knows what everyone's doing. And we need a way to like figure out how we're going to actually do projects and decide what we're doing and move towards products. And so that was really my motivation to come out of the lab and work more on how do we manage programs and how do people work together to do great science.
Awesome. Well, let's kick off with our first segment, when biology stops being science and becomes a system. This is going to be the breaking point between research and repeatability at the end of the day. So at Bolt Threads, what was the first thing that broke when you tried to scale biology?
And we can give a little background about what Bolt Threads was trying to accomplish.
Absolutely.
I think we've had our past guests,
give a little background. So
David Bressler, talk about it.
Yeah. So, following on other great past hosts of this podcast. And so David Bressler gave a good intro on all the things that Bolt Thread was doing. We were working on trying to make spider silk in yeast. And so we were putting the genes for spider silk proteins into yeast, getting them producing the protein, purifying that protein, and then spinning that into fibers, yarn, fabric, and apparel products as a demonstration. And we were able to, I was happy to manage the programs where we launched the first ever available commercially products made with spider silk. We launched a tie and then later a hat that was blended with wool, and really cool to see a biology product, but that's a physical good that you or your grandmother, anybody could have in their closet. And so in terms of scaling, I think I like to think about what do we mean by scaling? And so part of it is, how do we produce a lot? Of course, that's like the easy one, is like the obvious one is that we're scaling up production. And how do you scale production? And I think part of it is actually, sometimes we say things didn't scale well. We actually mean like it didn't make a good product. And so you can scale up production of something, but actually if it doesn't have the properties you need, then that's not going to be that do that well for you. So I think ultimately what one thing that we learned at Bolt was the importance of prototyping and making sure that what you're making actually tested as a product has the properties that are at least the minimum properties that you need in order to have the product properties that you're looking for.
So, what would you say broke when you were trying to scale that? I imagine going from like, silkworms producing
Mhm.
silk to doing it in this new process, there were a lot of pitfalls or hurdles that maybe weren't anticipated, right?
Yeah, there's uh, I mean, part of it again is just how do you make enough to make a prototype? So the issue with making materials is you need now like milligrams and then grams of materials to be able to test the properties of those materials. And so we're always looking for proxies of things that you can test at a smaller scale. And of course, those proxies are imperfect. And so there's the notion in high throughput screening of cliche, you get what you screen for. And so making sure that like the tests that we have as we are trying to scale actually are good tests that actually are predictive of the properties later on. So we definitely had cases where we're producing or even just trying to have a yeast strain that makes a lot of this protein and find that if you can grow it in high throughput in small volume and find that it makes a lot of protein when you make some modifications to the genome. But then when you grow it in a fermentation, which is a very different environment, it may act very different. And it's hard to scale between those things. And so making sure that the measurements are good proxies of what we're actually trying to do.
Yeah, that's a really good point. Thinking about like it is a proxy for a reason. It's going to
Right.
have its own issues, right?
Right.
Yeah, I was really fascinated with the Breslau episode where it was just, you know, to be able to accomplish that, and to be able to produce those materials, without the original article, just that alone. And I mean, yeah, you have to imagine there's all these other factors. And so, what's the moment where experiment becomes a system or fails to?
Yeah, but so my sense on the system is like when I went from academia to industry, the thing I jokingly said, maybe not too jokingly, is like in industry it actually has to work, and in academia, get it to work at least once or twice, and then publish as quickly as possible.
Document it.
Realize where it was broken, and then get out of it. Um,
It's an
but again,
academic exercise.
right.
Of course.
And it's great to publish our observations, but in industry, ultimately, we know it has to return value, so it has to really work. And so, um, a lot of what I try to do is like we don't want to say that things can fail because science is hard, but it should only be because hypotheses failed, which means like you came up with the designs or hypotheses you're trying to test. Those might be wrong, those might be bad, and then they can fail, and that's okay. That's the iteration that we want to be able to do well at. But there's a lot of other things that can get in the way,
Yeah.
like how do we have multiple people working on different parts of this at the same time? What is it that we're even trying to build, and is that a good product? And because I did a lot of project management is again just how are we managing these projects and being as efficient as possible, meaning like how are we taking as many shots on goal as we can so that we can do a lot of experiments, and many of them will fail, hopefully because because the hypothesis was wrong and we're going to learn, not because the design of the experiment was bad, we didn't learn anything, there was quality issues, etc. And so a lot of what I do is try to like build those processes by which we can do science and then go through that process to make great products.
Gotcha. Gotcha.
Yeah, that's super important. I think I've had so many conversations where you get pulled into a room and everyone's like, what are we actually trying to build? Because we've been doing this for months.
Yeah. What do you find that teams underestimate or make a lot of assumptions about? What areas, transitioning from bench to
Mhm.
system, they're kind of repeat offenders, if you will.
Or there's different considerations, right, on the
Mhm.
bench than when you're scaling to a system. So definitely.
Well, both. I mean, a lot of the things I manage are like research projects at the bench, and then even research projects that are part of scaling up to large scale production. There's definitely like there's the classic underestimation of how long things are going to take, how many iterations are we going to have to do. Just every time you're doing something new, you assume it's going to fail for I call it like unknown research demons. Like you just don't know why it's going to fail, and it might be again because of the hypothesis, or it might just because oh, we forgot that we should have changed that nozzle shouldn't have been open or whatever. Like there's just failures along the way. And so you really have to build that in. You can do, risk mitigation and like get in a room and think about all the risks that could possibly come up. And that can help, but still there's going to be things that are going to come up. So there's always this iteration and learning.
Yeah. I think you just touched on the next point, which is, what are the early decisions that can help something to scale? It's like risk mitigation. Do you have any others to add to that?
Planning.
Planning,
Uh,
good
you
planning.
know, uh,
Adding experimental
when I
buffer,
started,
like just in case.
yeah, well, certainly buffer, certainly make, well, I'd say two things. One is the planning. So like when I first started doing, like became more formal project management and did some training in that and learned about bringing in both traditional project management and agile that the people in IT were doing. You ask like scientists to plan and they go, I can't come up with, I don't know what's going to happen tomorrow, let alone the next 3 months. But there's always a way to make a plan. And like,
Yeah.
let's estimate, like, again, we don't know how many experiments are going to take, but we know how long each experiment takes, and so we kind of map that out. And then based on intuition, we think it's going to take maybe 4 or 5 tries. But if it takes longer, we have also a range of times that it's going to take, and we also know like the key milestones that we're working towards. And so, because again, this is like in project management 101, which is just like, what's the end? We'll keep the end in mind. Like, what is it? Where are we trying to go? And it can't be 2 years in the future because that's too long. So, like, where are we trying to go in 3 months? And so, as people are off doing their things, you come back in a room at some frequency and say, okay, are we still on track to this thing? Are we still marching towards that thing? Otherwise, like scientists are going to go off on their own, and they can go off in different directions. One and two, they can will also say it's never good enough. It's never ready. And so, like, what is that endpoint of like this is good enough to show that we've de-risked it or to make a product that's at least minimally viable.
That's so interesting you say that because I remember when I was in academia in the research lab, it was just a lot of flexibility and innovation at like a turn of a dime, right? You don't have the best equipment, right? Because we can't afford it. But when you get in industry, suddenly you have these project managers and they're asking you like timelines, and you're just like, I don't know. I've never actually like condensed all my madness to a
Right.
timeline.
Yeah.
Yeah.
But how do you plan a business without having at least some roadmap that you're trying to get to? And that's what we've done.
It really is a delicate balance for companies, to have this kind of free flowing idea. That's how things are created, new things are designed. A lot of new ideas come up, and also keep it within the bounds, and on a trajectory that leads to something that the
Within
company,
budget.
yeah, within budget, and that the company can justify and rationalize, the investment that it made. So it is a delicate balance, and PM can stand for project manager, product manager, or personality manager.
Definitely a lot of, I think that's the other point is like all of this is about the people, right? Like
Sure.
you guys talk a lot on your podcast about automation and all that. It all comes down to like the stuff that I'm working on is you need the people to be able to work together, to agree on where we're trying to go, and to ultimately like partner and get something there.
Yeah.
So PMs are really people.
People
Well, and
management.
then not everybody likes to do it too. So like, the people who like to roll up their sleeves and get a group together and be like, what are we how are we organizing is a skill that, not everybody likes to do. So I'm happy that I've had the opportunity to do it.
Yeah.
And you've got the unique perspective of knowing the science, and being able to kind of coordinate those conversations and those thought experiments that they're having into achievable timelines.
Yeah, I've seen the pain on both sides, I'd say, and I find that try to speak truth to power, which is on both sides. So like the people in the lab have a lot of knowledge of what's happening. The people who are executives have a lot of knowledge about what they want to happen and where they want to go. And like part of it is like, speaking to both about what is necessary. And yes, I know you want to launch a product in 3 months, but like, here's a path that could happen, but there's immense amount of risk on that. And so we shouldn't necessarily plan on that, but and then for the people in the lab, it's again, like, I know this plan sounds crazy, but let's talk about what is possible or what's going to be our best path forward to try to get to these goals that maybe were brought down onto you, but like, let's talk about what is reality so we can meet in the middle somewhere and get something done.
That's huge. And a perfect segue to our next segment, why most biotech never becomes a product. The gap between working science and usable product. And, sometimes, preparing people in the, when you go to a company, right, of preparing folks that, look, this is an industry where the failure rate is pretty high, and that's okay, right? It's just it's part of the deal. But let's take a little bit of a dive in that and your experiences. And so at Ginkgo Datapoints, and actually maybe you could give us a little background on Ginkgo Datapoints itself.
Yeah,
Yeah,
sure.
that's
I
neat,
know
right?
I know Ginkgo, but at a fairly 10,000 foot level, and a little bit down on the floor, but do specific automation. But
Yeah.
This is
give us
moving
some
back
insight.
from the racks, right?
Yeah.
Yeah, so we talked about when the last company I was at was Bolt Thread. So now I'm at Ginkgo Bioworks, and in the last 2 years, we started this group called Ginkgo Datapoints. So Ginkgo Bioworks has been around for I think over 15 years, mostly in industrial biotech space. So also working on yeast and bacteria to make either small molecules or proteins or strains themselves that go out and can do a function. And so doing that in partnership with tens or hundreds of companies at a time, so many projects ongoing. And in doing that, we built up a lot of expertise in high throughput screening, automation, and where I'm working now in protein characterization. So we were doing either enzymes or other kinds of proteins. And so we've gotten good at characterizing proteins. And so we spun out this, it's really still part of Ginkgo, but this business unit within Ginkgo Bioworks called Ginkgo Datapoints, where instead of trying to make a protein or a small molecule, it's really the data that is the product. We used to say we weren't a CRO, and now we're like, let's be a great CRO.
Okay.
And product though is really the data. And so that's why it's called Ginkgo Data Points. And so where I'm working right now is partnering really closely with many big pharma and biotech companies in the space of antibody characterization.
Okay.
So antibodies is a big class of drugs. They bind and they have function, but they also need to have other properties in order to not just be good binders, but also make good drugs. So make them manufacturable and stable in the body and things like that. And so we have a suite of assays that we do that are characterizing the biophysical and other properties of antibodies called developability. And so again, it's these like the other properties besides its main function. And what we do is we partner with, again, biotech or pharma, either as directly as part of their campaigns, either trying to make antibody drugs, or for them to make great AI models so that they can use those AI models to streamline future campaigns and ultimately do less screening in the future because they've got great in silico testing.
That is
That's
cool.
amazing. Yeah. So you're basically taking all the data that either they're generating or you're generating.
Mhm.
In the case of the AI models, that the more that you have, the better, right, for the model. And
Right.
then in the case of actually trying to go towards, a viable drug, now you have the backing of all the experimentation that you are sort of in the middle of, right?
Right. Yeah. It's again more data is better is what people say, and also really now having done this for a couple of years, we've learned like not like which data is useful, particularly for model training. So you want to
Mhm.
have a diverse set of inputs, both in like the sequence of the proteins and also the properties that you measure to be able to then to predict when the next set of antibodies come in, what are those properties going to look like?
Do you find that a lot of the data that you're looking at is viable, something that can actually be used?
Yeah. What's important here is that the data is good, not necessarily that the antibody is good. Begin because people want to know what is good and bad, and
Yeah.
and the model needs to know what does good and bad look like. Because if you only give it good proteins, it's never going to be able to tell
Good
you what
point.
that protein looks like.
Yeah.
And so, what's really important with the data being the product is that the quality is really important. Something it's always important. But if you're doing high throughput screening, I found that you can be a little bit lax because the important thing is finding a hit. And so as long you can have some false negatives where like you may have missed the good ones or even false positive. Ultimately, it's did I get enough hits to make a product? And for us now, again, like the data is the products. Really, a lot of emphasis on quality, making sure SOPs, of course, is necessary, but also that we've got controls. We're monitoring those over time. We have acceptance criteria. We're actually rejecting runs of methods if they haven't met those acceptance criteria, so that you have faith in the data. And then it's a matter of like, again, experiments can pass or fail, but are you confident in that outcome more so than did you meet your hypothesis?
Yeah, that's 100%. I love that. And, protein characterization is close to my heart. I came right out of school into protein characterization in industry. So it's great that you're looking at all that.
Yeah, that's it's just been amazing to watch this industry, in general, like research space, where the new companies are becoming essentially biology software data as a service to support the overall industry.
Yeah.
It's like the challenge has gotten too big to just keep it all under one roof and do it all yourself. I think that sort of answers the question about productizing biology and productizing for a company to be able to provide these services as a subscription. I assume it's like a subscription model, or is it like a particular, like a project by project kind of thing?
Uh, the project has been typically directly with the companies.
Okay.
We've partnered with, I think now 8 of the top 20 pharma companies and other small biotechs. And so far, those projects have been one-on-one. So they're coming with sequences that they want to have tested again, either as part of a campaign. So like they have a lead and they have variants on those, and they want to see how that's going, or they're designing a diverse set of inputs again to be used for modeling. And so typically, so far it has been a one-on-one, but we're also finding that the industry is very hungry for models, and they know they need more data. And so we see at conferences that I go to, people saying it'd be great if we could find a way to collaborate across companies. And so we're in the process now of, I've been asking like, well, how do you actually, everybody says that at a conference and then they walk away. And
Yeah,
wait,
absolutely.
Next.
Yeah. It would be awesome if we do this. I'm like,
yeah,
somebody
like,
has to start.
yeah, how does it take the effort to do that? So there have been some efforts where people are combining historical data. So like the point there was like everybody has data, so let's find a way to combine it together. The angle that we're taking is we can do some of that, but also we can pool resources and generate the new wish list, largest ever, most diverse data set that's going to make the best models. And we can do that by bringing multiple companies to the table. And that is not easy to bring multiple companies to the table.
True.
Not only do you need to have lawyers agree, but you need to have scientists agree on the methods that we're going to do. But we're in the process of putting that together. We've got some good pharma partners at the table and hoping to launch that later this summer. So a consortium of companies that are all buying in together, and they'll all have access to the data, those that buy into it, and then they'll be able to. We also have a team of modelers, so we'll deliver a model on that for them to use, and they can use the data to train their own models. Some of that through a federated network, and so
Okay.
some of the sequences will be sourced from the pharma partners, and they don't want to share their sequences with other companies. And so there's ways for the data from each of those member sequences to be stored in a central place through a federated partner. We're working with Ferris,
Hmm.
who's out of Germany. They put together a bunch of these networks already. And so the ability for everybody can model on the data, even if you don't see the data directly. And so you benefit from now having 10,000 data points, and then you can go and use that model for your future campaigns.
I love that. We've been talking about trying to see if companies would ever come together for something like that. This is great.
It's beautiful to see. Yeah.
Yeah.
Yeah,
A step forward for sure.
definitely.
Yeah.
Definitely. Yeah, it becomes, a daily kind of thing, to work together at
Mhm.
that level between companies. So that would be
And to be able to
great.
model against it without, worrying about
Yeah,
IP.
Yeah. No, we think a
exactly.
lot about data security and stuff and making sure that that works for that the data is secure and that people don't see access to other people's data.
Yeah.
Also understanding again, like how are they going to use this? We're spending a lot of time talking about how are you going to use this model? How do you justify it internally for the investment? Because you have to have a return on investment to make it all worthwhile.
So touching on this, the segment title, why most biotech products never become a product, or a biotech experiment. Where does the friction show up? Is it in the data, the workflows, the people,
The
the
integration.
integration? The
All,
everything, all the above.
I think all of those things. I think again, it starts with a product, and so like keeping the end in mind and making sure. So some things don't fail because either you weren't designing towards a good product, or you didn't even think about that, and so you're busy in the science, but you didn't think about
Yeah.
the product. Back at Bolt, I was working with a lot of people from Nike because we were making a
Oh,
video
right.
game.
Yeah.
Ooh, that would have been cool to have a shoe. Yeah.
Yeah. Well, I mean, but I learned from them a lot about product, design and product management and what is a product brief. And so every time I'm working on, either an apparel product or now antibody characterization product, it's always like, again, what is the thing that we're making? Who wants to use it? and how do we make sure we're designing towards that? So that's the starting point. And then I think some of the failure again is that you have to then you have to de-risk some things as you go along the way to keep management continued to be interested in it. That like, either not that you necessarily run out of money, but at some point management goes like, oh, that's not going fast enough. I'm going to move to something else. So you have to be able to show along the way that, look, I'm telling you in 3 months, this is a big de-risking thing you should look for. And now we're marching towards that. And okay, we've achieved this goal. So maybe we're not at the end point yet, but like, this is why we've worked out some of the risks along the way. And we've demonstrated, partially towards what the goal we're trying to get to, and let us keep going forward. And so you have to keep going forward before you run out of money.
Yeah.
Or get pulled onto a different project.
But I mean, running out of money doesn't mean the company runs out of business necessarily. It's just the management pulls the money somewhere else.
Yeah.
Well, and another arena around, balancing between what the research folks want to do and management, right, is, how do you balance scientific flexibility with product standardization?
Back to, I was mentioning like a blend of agile project management and traditional, where traditional is like a Gantt chart. And I do love a Gantt chart. And so, like what is
Quote of the day.
right. But you still need to have like, rapid iteration on a shorter time frame. And so I like to have like a blend of those. And so like, what experiments do we have live right now? When are we going to get results from those? And then we can act and do the next thing. We're still working towards that, like maybe 3 to 6 month goal, but we can iterate on shorter time frames.
Yeah. Agile was a fascinating, I was sort of on the sidelines, doing the clinical development of a medical device, but our R&D team was full on agile, and our week to week clinical trial iterations were just immense. Like, it was amazing. Like, we were moving so fast. Now we had to kind of rein things in, but I'm with you, that balance between Gantt chart and agile, they sort of both have a play, I think, in reality. Yeah. Well, so switching gears a little bit to the reality of AI and data and antibody discovery.
Mm-hmm.
Right up your alley.
Right. Taking further, deeper dive. So where is AI actually delivering value today in antibody discovery?
It is definitely being used, I'd say, across all pharma and biotech right now. It is, it's either they're using it or they have to move towards it.
Mm-hmm.
And that is part of it is because of hype, and we have to move past that. And that, like, what is the reality of what it can do? So, like, again, there's hype in that, like, I want to have a model because Amgen has a model and AstraZeneca has a model, and that's not good enough to just want a model. It was like the South Park thing, like I'm going to buy wrenches and then I don't know what next at some point.
The South Park.
The places where it's succeeding, and I'm seeing a lot of talks from some pharma companies who are really invested in using it to change how they're doing their work. And so the AI models are only good enough if you're gonna, especially if you're gonna, you're gonna invest, you're gonna have a team to either generate data or make these models or buy it from somebody else. And to get that return on investment, you have to say, okay, now how am I going to use it?
Mm.
What is the return on investment? And it comes from having those models to do in silico screening so that you're doing less testing, or that you're committing, you're moving better things forward so you have a lower chance of failure as you go along. But anyway, it takes like going from like a traditional way that people are doing things and saying, okay, we're going to actually do things, we're going to do some transformation to do things somewhat different. And that requires some change. And I know that these old pharma companies can struggle with that. So the ones I've seen be successful are ones who are really committed to transforming how they operate to get the return on investment. And the thing about, like, I'd say the hype and reality is that the holy grail that people want is like the de novo design, where you can walk up to a computer and say, I got this target in mind. If only I had an antibody against this target. And then you walk up to the computer and you dial it up, and now you've got a drug that you can take to market. Reality is that there's always going to be testing involved. And so really it's just a matter of like these incremental benefits that we'll get along the way. And that's what I'm seeing is that you still have to test, but you're testing less, or you're failing less, or you don't have these campaigns that are dragging on for months and months because of some issue, because you've been able to do some in silico screening earlier on. It's not quite as like sexy as like de novo. I walk up and I can design
Yeah.
an antibody, but just like these incremental improvements that is really going to change the industry.
And we've heard that before from our guests on the AI series. The incremental improvements are more significant than this holy grail end goal. Even when automation first came out, right? No one ever just went all in and said that, like, we're just going to trust this completely. There was always testing, benchmarking against like what you're doing manually, right? And so, to your point, testing is very important to the process.
I've never seen a technology have such a prolific impact in an industry like AI and ours, but there's still going to be, the overhyped or the, not quite there, as you were saying, that conversation about incremental improvement it just keeps going and going and going, and people are able to get better and better results out of their use of the technology. But what are some areas, Rich, where AI is still not living up to expectations? It's one of those things, like we heard about it. This is where it should be used, but it's just not living up to it.
So interesting. We ran a competition last year in the space of developability, and so again, a lot of people are doing trying to design binders, and so like how do proteins bind to each other? Even before AI, there was a lot of computational design for this, and there's Nobel prizes now on how to do it. Not to say it's solved, but some people say depending on the antibody format, like coming up with good binders has made a lot of progress, and now it's predicting these other properties. And so again, things like how much is a protein going to aggregate, how stable is it going to be at different temperatures, how polyreactive is it? So like yeah, sure, it binds to your thing, but how much does it bind to everything? And all those things create problems when you actually try to make a drug. And so trying to predict those properties from the sequence of a protein is still a challenge. So we had we've put out a data set of 250 antibodies, which is big, but actually relatively small to like how much people are actually going to need for modeling these kind of properties. And then we said, okay, we've tested 80 more. Here's the sequences. We're not going to tell you the answers, and you predict the answers based on all the models you have from all the data you might have generated. You can use our data that we have put out there, and then submit your guesses, your predictions for the properties of these proteins.
That's so cool. A binding hackathon.
Wow.
So it wasn't binding. So it's actually like these other properties.
Yeah.
Oh,
Yeah.
okay. Yeah.
And so these, I think there's 5 or 6 properties that we put out there, and then we evaluate the submissions and we look at how good do they do at ranking them. And most of them were pretty poor. Yeah, they weren't great at predicting the properties of these sequences. And so it's a bit self-selected. It's like we haven't tested every model in the world. We only tested those that were submitted, but we got submissions from all over the world, and we got hundreds of submissions, and like most of them were pretty poor, like not good enough to actually make an impact in pharma. And so it's there's room to grow, and so we do think that, like, again, we need better models. We need more data. Of course, we're biased. We would love to generate that data, and that's what we're doing with various members. And so this global generalized model for these kind of properties are not yet there, but we're making a lot of progress over the last couple of years, and should expect so for the next few years.
What inspired a competition?
Some other people have done that. For protein folding. There's the competitions that have been going on for 20 years or so, where people they put out a protein sequence and they say predict the structure of the properties, and that's where AlphaFold over the last 5-10 years has kind of demonstrated, wow, this is like much better than other things. And so we at Ginkgo, even before Datapoints, we've always kind of wanted to be also community builders, and so we try to be people who are trying to bring the community together to assess where are we and how can we improve from where we are today.
Very cool. Yeah, we had Ingrid Barbosa, and she did a deep dive on, for a 1-hour podcast on
Molecular docking.
Yeah, molecular docking and AlphaFold.
Yeah.
That was very cool.
Awesome. Yeah.
So this is what I've been looking forward to asking you, because there's always 2 sides to the perception versus the reality, and I love that kind of inquiry. What do operators understand about data and AI systems that the market just gets wrong, just completely? It's not how it really works.
What does the market respond to? The market responds to not just hype, but like outcomes. And so, really seeing actual, impact being made by these things. But the market also responds to like new partnerships. And so we see stocks move all the time just based on we started a project with somebody.
True.
And And somebody who likes to execute and deliver, really it's all about delivering on a project. And so I think the operators might roll their eyes and go, "Oh, great, hey, more work to do. Great, we signed on the thing." And great that the market reacted to that. But and we can have a huge dollar sign because there's all these milestones associated with it.
Yeah.
But the real success ultimately is based on how much impact did you have, how much did it generate revenue for your company and business. Jason Kelly, our CEO, always quotes somebody. I'm sorry, I'm going to miss who he's quoting, so maybe you can look it up. But that the market in the short term is a voting machine, and in the long term is a weighing
Oh
machine.
yeah.
and and so ultimately, again, it's about outputs. I'm not sure that you asked though about AI. It's similar. It's again
Well,
like
yeah. I mean,
using AI,
yeah.
but the reality of like where are we today? Again, I think we've made a lot of progress.
But
Yeah, but
like you
that's a
said, the
good
outcome.
distinction,
Yeah.
right? Because the people doing the thing have a different perception than the market, because the market is like, oh, what's sexy? What's going on? What's going to bring the most hype, right?
Hmm.
Yeah.
Yeah. So we like Ginkgo announced we did a collaboration with OpenAI, and very cool. We used our very advanced automation systems we have here. We call racks, and we're building a autonomous lab where the racks can all work together. And the collaboration with OpenAI is to make to let the AI not just analyze the data, but actually design the experiments. So read the literature, design experiments, execute the experiments, then react to the data, design the next experiments, et cetera. And so, the AI scientist is doing that work. And the reality, if people can actually be, I think some people look at it and they're like, wait, but humans in that experiment were actually reviewing along the way
Mm.
all the steps and like, yeah, and actually Ginkgo has put out something we call the levels of autonomy. And we acknowledge that we're not at the point where like AI can really just do this on its own. We never have to come to work. That we're at a toddler level of maturity where we are putting together systems that can do like a lot of the stuff that we could do on our own, but that we're still influencing it and having checks on it. So I think the reality is like the OpenAI collaboration is great, and it's like a step in the right direction. And if you can look at the paper and you can see that same thing, and so people could be critical. Oh, humans were involved. It's like, yeah, okay, we're making progress, but
Yeah.
like
Less and less, but still
humans are
involved.
always
Yeah.
going to be involved. Actually, it's about the people.
Yeah.
And we've heard that throughout the community talking about AI, right? All the thought leaders are saying AI is at a toddler's level of maturation. Like, don't expect that it's going to be completely not backed by humans, right?
Right.
Yeah. Yeah. It's a tool.
Yeah.
At the end of the day, it's a tool when we
So
use it.
definitely want to link that article. That sounds interesting.
Yeah.
Uh, so that I think leads right into this one around, okay, clearly there's a business around using AI for the models, right? And building
Mhm.
models. But what about other ways of integrating it into lab and real life workflows, and enhancing things we can do today to enhance that? Where should teams be looking to integrate that?
Yeah, so we've been talking a lot about like how AI is doing science and how AI is used for modeling, but we're using AI like everybody else, other industries in terms of just like basic workflow. A lot of people here are using Claude to say, okay, I'm going to code a thing that's going to make it so that every time I need to make a new certificate of analysis for a method that we just put out there, that like that can be made more automated. Everything can be made more automated now, and same with like drafting contracts and drafting. Uh,
Yeah,
uh,
it's awesome
you know,
tool. Yeah.
you say like, here's the basis of what we're trying to make, and then work with AI. We found it best when you kind of do it conversationally, and so like give me some ideas of how this should be formed, but ask me some questions before you go ahead and then write the first draft. And it actually does a very good job. Actually, one cool use case was been doing all these joint meetings with the multiple companies through the consortium. And we find that we have the AI notes. So like, I think the entry
Mhm.
point is people like, take notes from this meeting. It does actually really well. And so every time we have one of those joint meetings, I can send out those joint notes to everybody. And now we've got notes from 8 meetings, and we go, okay, actually summarize these 8 hours of more than 8 hours of meetings that we had into like, okay, what's the consensus? What does each company think that we need? And how do we make sure we incorporate that into the plan? And it's just hard to remember all that stuff. And so, taking notes on paper is the old way. And now like taking notes with AI has been very helpful.
Especially when you want to pay attention to the meeting, right?
Right. Yeah. Yeah. It's been a very interesting exercise in facilitation.
So heading into our final and fourth segment, but what everyone gets wrong about scaling biotech, and this again, I like these kind of a little bit controversial. What is the most overhyped platform in biotech?
I think again the idea that de novo design is the only use point is overhyped, and so again these kind of incremental things are going to be important.
I really liked that,
Yeah,
when you spoke about that. I think the de novo design is something that people are just like, "Oh, I really want this like yesterday."
and if it can't be if we can't do it now, then like I told you, AI wasn't going to work. Or I mean, I see
You
it.
went too far.
Like I've seen hate pieces about certain companies and be like, oh, they said they were going to do de novo, but this protein they put out there is just an improvement of another one that was already out there.
Yeah,
Still an improvement, bro.
it's kind of the wah 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 comment. Yeah, okay.
That's like you've got like all these fully electronic cars and people are like, they said we were going to have hovercrafts. And you're like, but look.
Back to the Future 3. Come on, I wanted the hoverboard.
Yeah.
All right, well, on to our next question. Why do platform companies struggle to consistently deliver value?
Quote unquote.
Whew, yeah, that's a good one. And because there's always a balance. Like at the first company I was at, we were making products, so we were trying to make apparel products, and we were keeping that. And Ginkgo has always been committed to the platform, and so we are going to be a platform, and we are going to enable people who are making products to make great products. And so we're gonna have the greatest platform, so you're not even gonna need a lab anymore, because you can do a lot of your research with us. We're gonna make biology so easy to engineer that that again each one of you will be able to make great products. And along the way we have to think about, okay, how do we what value do we get returned? And so how do you price that? How do you put value, particularly in early research, where someone's like, I get that you're adding value to my early research, but there's many other things that have to happen downstream, and so I can only put so much value into you helping with research. And so we've done a lot of like trying to come up with different pricing models. Well, you only pay if you can actually ultimately get success on it, or once you put a drug into the clinic. Or now again, like it's not even just based on us delivering something, but you get the data. So actually just what Datapoints is doing is, your question early on was, did we make a great antibody? We're actually focused again on just like we're great at characterizing the antibodies, and so that's the return. And ultimately, I think you need to figure out again, like how does that scale? Because services are hard to scale as a business. And so then again, trying to think about autonomous labs and how do we do this more and more so that they to do 50 projects instead of 5 doesn't require just 10 times more people. And so how do we become more efficient? And so you have to come up with some way that we're going to be able to either become more efficient as we do more, or to make a product, or to get return on a product. And so I think the platform team struggle because ultimately, like, the value is in the product.
Sure.
And so it's hard to translate it back to how much did the platform create value in my ultimate product.
Yeah, good point. How do you get that metric, right? If you've got that product as the end. Yeah.
And we also now have automation as a product. And so we've got
Yeah.
really awesome labs here. And so we're kind of, again, we've been exploring different models over the last 15 years. I've been here for about 6.5, and so I've seen a lot of different things happen. And now part of it is like we're doing all this great work on this automation, and it's really reconfigurable and standardized. And so continue to work with us as long as you want to. You don't need a lab, but if you do want a lab, you can buy this product. You can buy the automation. And we can help you get it installed and even help you operate it. And because, like, it's like these carts, and you can do the same thing in your space too. And so again, trying to find different ways that we can recoup value for the work that we're doing for people.
Very unique level of vertical integration over
Mhm.
there. Yeah, I mean, just all the way down to the hardware, which is really rare in this space to
Yeah.
cover it from.
Great episode from Will Serber also on your
Yeah.
podcast.
No, we had a lot of fun with Will. Yeah, exactly.
Rich, you're hooked into everything. David Bressler, and then we've got Will Server.
Yeah.
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 And I think you touched on this a little bit as far as like the underestimated part, about executing at scale, right? Going from the 5 to 50 without going from 5 to 50 people, people wise. But anything else you wanted to add to that as far as things that are underestimated to scale up?
Yeah, I think again, what I used to call more traditionally scale up is like we're producing something at a 1 liter scale or a 1 milliliter scale, and then we have to go to 10 liters, 100 liter, 1,000, 100,000. What technologies scale well? So like when I was in the more synthetic biology space, many things are going to want to scale to like really large tanks and really large purification. And need to be thinking about how those things scale. And there's a lot of science in how things scale, recognizing some things work at small scale that don't work at large scale. And also recognize that there's going to be some failure and iteration. And so, I mentioned earlier the story of like executives want to go fast. Like, okay, we're going to have this new facility opened up. It's possible it could be ready faster than any facility's ever been run, and we're going to try to make that the case. But reality is, there's just going to be some tripping on our feet that we can mitigate risk as much as we can. But it just takes time to onboard anything new, and so anything new is going to take time.
Yeah. Yeah.
Those are the meetings that you're like, I'd love to meet that timeline and I will try to meet that timeline, but it might not meet that timeline.
Right. I'll have a better sense in 2 months. So let's meet again. And yeah.
We just integrated this software into this company 2 weeks ago. What do you mean? We got to work out the kinks. If you were starting a biotech company today, what would you do differently?
I like to join a company and execute. So I think it really is, what is the product that we're trying to make? And also, who are we serving? Whose problem are we solving? That comes from being a CRO service or a physical good that you're selling to people. And then iterate, because you're going to be wrong on that too. Like everything is an experiment. And so what I learned I didn't appreciate when I first came into industry, of just that something has to work and you have to show that you're de-risking something. So again, I think it would just be making sure we understand what problem are we solving, where are we differentiated some on the business side, but also then like understand everybody who's working on it, even down to the operators, they understand that the use case of the thing that we're making, that we iterate on that, because we're going to be wrong and the markets are going to change and everything else. And the science is going to take us in interesting ways. And we might find that, that we learned that actually this thing might be really well suited for something else.
Sure.
Let's continue to iterate and go. So I think it's always really appreciating that from the get-go, is understanding again, whose problem are we solving? Oh, I just also want to really appreciate operations. And so shout out to operations, which I know you get a lot of people on here on other podcasts, again, talking about the hype and the vision and all that stuff. And so I want to give a shout out to like the people who execute and operate, and they don't all have to have PhDs or anything. It's really just about how do we build a really good process.
I love that.
Shout out to ops.
Shout out to ops.
By the way, shout out to the hype and the vision people too. We need all types, right? Like
shout out to the dreamers.
We need you both. Hype it up. Execute, hype, execute, hype, execute, right? It's the cycle of business. And
look at Rich including everybody in this.
Right.
As we bring it in for a landing, we always have this lovely segment known as the pick of the week. And it's flexible, right? Do you have a framework, idea, a resource, a tangible item,
new gadget
shaped how you think about building systems in biotech or in general across the board?
Yeah, so haven't watched now a few episodes to prep for this.
It's not a surprise anymore.
It's not. Well, I appreciate it that you guys have this segment. So, and I actually watched a couple to see, like, doesn't necessarily have to be an AI tool. So I'm going to give you one that's something that my wife and I just finished watching, a show called Company Retreat.
Ooh.
And I don't know if anybody saw Jury Duty. It's both by the same people on Amazon Prime, where they have this concept where in Jury Duty, it was everybody gets called to a jury and one guy doesn't realize that it's a TV show. And so the whole thing is a premise where like it's crazy things happen and this one guy doesn't know.
That was hilarious.
It was very good. So they have a sequel now called Company Retreat, and they've got a guy who signs on as a temp to support this company on their company retreat. They go away for 5 days and all these crazy things happen and the company almost gets sold. And I'm not giving any spoilers away to say like he's the one guy that doesn't know that it's a show. And it's very entertaining. But also the the kind of takeaway from that one is like they build these bonds really quickly in these like 5 days that they're together, and it's really about like the people of an organization. And so that's one aspect of it is, again, it's like the people trying to put out a good product with the quirks and their own things going on in their lives and all that stuff. The story comes together like this guy really interacted even in 5 days, like however many days it was, becoming really close with these people and connected to the mission and what they were trying to do. And the other aspect is like the amazing challenge of the project that the producers of this show have to go through to be able to like film for a week without this guy knowing that somebody knew he knew there was a documentary, but there's like way more cameras and like dozens of people that he can't see, and all these improv actors are like been like making a plan for this for weeks. And so it's a really amazing project too, in terms of how they're able to pull this off. So,
And that's a bigger scale. Jury duty, at least you were like in rooms,
right. They
Yeah.
point that out. They're like at one of these, you can imagine going to a retreat with 15 people in some place in California in the mountains. And yeah, it's this grand scale. And so amazing that they pulled it off. Amazing that it kind of went along to plan.
Wow.
But who was duped along the way has a great spirit about it and is now a famous guy, I guess. And yeah, really excellent show.
Oh, I want to watch that. I watched the first one and I was like, oh, this is amazing.
Yeah.
These producers and writers, you think that they've come up with everything. There's always something around the corner.
Well, so imagine having a script where one person doesn't know the script.
Yes.
Everybody else does.
That's cool.
And he's the star.
Right.
Jeez.
Right. Oh, that's great. That's great.
Yeah.
And you also have a new microphone.
Well, I don't get to take this one with me. Ginkgo does a lot of, or Jason Kelly does a lot of like podcasts and stuff. So I borrowed a
Oh,
new
great.
microphone.
Yeah.
Like, so, yeah.
It's a proper microphone. Yeah.
Podcast circuit if anybody else is listening.
All right.
It's not your debut. You did one 15 years ago. Yeah,
Ladies and gentlemen, Rich Crone is available for podcasting services at your nearest, convenient
Please
time.
contact him for bookings.
In the show notes.
It'll be in the show notes, and that, yep, and we've just got to fit it in there. Smash that like button, all right.
Smash the like button.
Subscribe today.
Sura, pick of the week. This is also very famous too.
I don't have
Yes. So we want to make sure we cover things. I do have a pick of the week, and it's an Apple product, MacBook Neo. This is $599. I went for the $100 more expensive version just to get a little more SSD, but it's actually running on an A18 Pro, which is an iPhone chip. So you now have a laptop that is powerful enough to do Final Cut editing that has a chip that's in a mobile phone. And it's not even the latest mobile phone. That's the A19. Anyway, that wraps up our pick of the week segments. And where to follow you, Rich. So listeners, you can follow Rich Cohen and explore what's happening at Ginkgo Datapoints. Where, sir?
LinkedIn?
Yeah, we do a lot of posting on LinkedIn.
Okay.
But I do a little, and Datapoints does a lot, and Ginkgo Bioworks does a lot. And so again, a lot about what we're doing in Datapoints. We're generating data both for, I mentioned the antibody product, but we're also doing small molecule, similar ADME characterization, and also functional genomics, transcriptomics. And so working again with connected into this biotech pharma community, generating high throughput data that really helps move the needle for them.
Very cool.
Awesome. Well, listeners and viewers, we want to hear from you too about this episode and other episodes. So what actually scales in biotech science or systems? Drop your take below and tag us with hashtag Helical Brew. For those that are listening to this on one platform, we do have others, and I'll go through all of them. We've got www.helicalbrew.com. It'll take you straight to our YouTube channel, where we've got a variety of shorts and full episodes available, and on Spotify, iTunes podcasts, Amazon podcasts, and TikTok and Instagram. But full episodes are all available on those podcast syndicators. So take a look. We're going to be out there. We're going to be doing more episodes coming up. And we want to thank you for joining us here on Helical Brew with Rich Cohen. Thank you for joining us, Rich. Pleasure to talk with you and have you on the show. And for now, this Helical Brew is brewed.
Hello.
Thank you for joining us on the Helical Brew. We're glad to have you with us. For more episodes, please visit our website at helicalbrew.com. That's helicalbrew.com.